structure and function, which entails the structure of the membrane itself. What is it made of? It's made of some lipids. We may have some proteins involved. There are carbohydrates attached to some of those proteins, which we talked about last time. And then what is the membrane's function? Its primary function is to act as a barrier from one compartment to another, but it also functions to facilitate moving things from one compartment to another, whether inside of a cell, as in a eukaryote, or between the cell and its environment in prokaryotes and eukaryotes. Okay, so if you look at a biological membrane, in essence, it's just a barrier that keeps an area of volume, or a region, a volume of liquid, separated from another volume. Okay? In the simplest terms, that's all it does, is prevent things from moving from one place to another. But it can't just prevent things from moving, it also must facilitate moving some things from one area to another. So there must be a way to get across this membrane. So we call these membranes semi-permeable, meaning some things can go through them, and some things have a more difficult time, and some things are disallowed from going through completely. So we'll look at what things can flow through a membrane easily, what things cannot, what is the membrane composed of, and how does this allow these differences, as well as can we change the membrane or use it for something. Okay, so the first thing I want to say is, what is it made of? So we like to call it a lipid bilayer, and for the most part, it's the bulk of the membrane. May not be the most massive part, but it's the part that functions mostly as a membrane, as a barrier. So if you imagine our phosphatidyl molecules from last time, where you had the glycerol with the two long fatty acid tails, and you had a phosphate on the other side on the third carbon, and attached to the phosphate was some alcohol group, and it varied, and it varies among membranes. So if you look at those at the very top of this figure, you'll see how several of these, in fact 6 of those diacylglycerols with the phosphate and the alcohol on them, are put together. You notice all the long tails tend to face each other. They may or may not be the same tails because you can put different fatty acids on, and that can play a part in how this membrane attracts certain proteins or lets certain things through. And the other part we're going to describe is not just the membrane itself. This is how it looks at the top, and then it's redrawn as the little ball and stick cartoon here with blue. And then embedded in that membrane are various proteins. Some of them cross the membrane completely, as shown in a couple of these examples. Some do not. They just sit in the membrane, right? They're not completely crossing the membrane. For instance, this one over here. Right? And then we also have other molecules in the membrane. These large pear-shaped things are going to be cholesterol molecules. Okay? So there's some proteins that go all the way across. How they get across, there's a couple ways you can do that. We'll talk about those in a minute. Or you could not go all the way across, like this one in the background here is embedded in the membrane, but it's not completely going all the way across as far as we can tell. And then there's some that just sit on the surface, whether the interior surface or the exterior surface. They don't actually embed themselves in the membrane, they're just associated. We went over these once before. We had the integral membrane proteins and then the peripheral membrane proteins. Attached to these lipids may also be carbohydrates, as in these green sugar trees. They may be attached directly to the lipid itself through the phosphate, or they may be attached to a protein that's embedded in the membrane. And we saw this last time, where we can have many sugar trees on the surface. In the interior surface, we also have attachments to these actin filaments that hold the cell together. And along these filaments may be molecules that act as transporters, and you can quickly get from one side of the cell to the other by following these tracks. These cytoskeletal elements, microfilaments or actin filaments, may also serve to move the membrane. You can tug on one of them and it'll change the shape of the membrane. This is how cells can move. And what holds this membrane together? Well, we know lipids don't form bona fide polymers, but they do associate with one another. And why would lipids do this? And it's to exclude water. So if you have water buried in the center of this lipid layer, it would have to be very organized, very structured. It does not want to interact with those lipid tails. So it's very limited in the orientation it can take. However, if you exclude that water molecule and push it out of the membrane and allow the membrane to reseal behind it, which it will, right, that water is now free to do whatever. Right? Its degrees of freedom have increased tremendously. We say its entropy has gone up. and an increase in entropy is a favorable process. So the reason the membrane forms a stable structure, this lipid bilayer, is because it's in a water environment. If it were in a lipid environment, like in hexane or benzene, it would not adopt this structure. The tails would be pointed outward and bury those polar head groups. Right? But it's in a water environment, so it adopts this structure, and it maintains it by excluding the water, and energetically the entropy of water goes up. That's why it maintains this shape. If I were to punch a hole in this membrane, the lipids would seal the hole. Right? They would close in on themselves to exclude the water from their presence, right? For the same energetic reason. So the membrane is self-sealing in that if you put a hole in it, and of course things can leak depending on concentration gradients and so forth, but the membrane itself will eventually seal the hole, or seal the barrier breach, because it's more energetically favorable to exclude all that water.
If you poke a hole in it with a detergent, why does that just destroy the cell instead of the membrane closing?
Okay, so when I say poke a hole, you have a good point. When I say poke a hole, I mean physically, like remove the lipids in that region. Not by adding another molecule or anything, just physically they just instantly don't exist. You know, like a plunger goes through it or something, right? A really tiny one. In the case of adding a detergent, what it's doing is a detergent or a soap molecule has hydrophobic and hydrophilic characteristics. So it interacts with the lipids, but it also interacts with the water and kind of leaves the hole open, which leads itself to destruction. So I'm just talking about physically, like using a hole, like a hole puncher you use on a piece of paper. Just punch a hole in the membrane physically, and nothing else added or in the way, it will seal the gap. But if you put something in the way, like a detergent, you're going to disrupt that ability to reseal. Okay. So what can we use these properties for? Let's say you have a solution here, and I'll just use glycine as an example, but we're going to see how this leads to other molecules in a minute. But I have a solution with, it's in water, so there's lots of glycine molecules floating around, they're very soluble in water, and I have a lot of these phospholipids in the same container, or they're not soluble in water, and they're more dense, so they'll settle to the bottom in this example. So I have my little beaker on the left where all my phospholipid in green is settled to the bottom, and the glycine shown in the red or pink colors floating around in the blue water. If I use a device called a sonicator, which is just a metal tip that emits high frequency, very high frequency sound waves, it'll disrupt the whole solution. It's kind of like stirring it up with a spoon, but you're using very, very, very tiny spoons all throughout the solution at once. So it's very energetically moving things around. That's all this does. Okay? Those lipids or phospholipids would rather associate with themselves than with the water, and given the right concentration, they will form little things called micelles. Right? Given a different concentration, they'll form a little compartment. Right? So if you have a low concentration, the tails will just associate with each other and it'll make a little ball. If you get the concentration a little higher, but not too high, they'll associate in a way that makes an inner compartment. Right? So you make a bilayer that's a sphere inside of a sphere, if you think of it that way. Right? So what we have is an opening in the center, and then a bilayer that's closed on itself. And remember, this is just a two-dimensional drawing, but this is also occurring in 3 dimensions. So it's a sphere where there's a space in the center that is now separated from the outside liquid. Okay? And if you imagine when I'm sonicating this thing, or mixing and stirring it all up, some of those glycine molecules, which are rather small, will get trapped in this inner compartment. Okay? So I end up with a solution that looks like this. If I let this sit for a while under gravity, or I filter it, and all the glycine molecules will fit through my little tiny filter, but the big circular or spherical lipid vesicles will not, then I can filter out all the glycine that was not trapped inside of a vesicle, and then I can shrink this volume down through evaporation or filtering again, and I can use this as a delivery system to get glycine into a cell, for instance. Well, that's not such a big deal for glycine, but if we replace glycine with another molecule, like a chemotherapy agent, or some other drug you want to get inside of a cell, and that drug is very water soluble, it's not going to go through the membrane rather easily. Right? We have a problem here. If a drug is very lipid soluble, it easily goes through a membrane, but the delivery is difficult because you can't put it in solution. Right? It's not water soluble. The opposite scenario, if your drug is very water soluble, It's easy to get in solution, but it's difficult to deliver to the interior of cells because it can't cross the membrane. So here's a way to get some of those drugs that have no other means to get inside of a cell. You have a water-soluble drug. You put it in a container like this. You sonicate it, and hopefully that doesn't destroy your molecule. And it gets trapped in these little vesicles. You concentrate them down. You apply this to a cell membrane, and some of them may fuse with the cell membrane. And your cargo, which is in this inner compartment, gets delivered to the interior of the cell. Okay? It's not 100% guaranteed, and it's very inefficient, but it can be done. Right? And of course, depending on which phospholipids you choose, you can change the rate at which this is absorbed by the cell. Right? So what things can cross a membrane? We said if it was very lipid soluble, it could cross a membrane. And if it was very water soluble, it cannot. Well, there's three main reasons something will or will not cross a membrane. Right? And you can probably come up with these on your own. If something is huge, right, a very large molecule, like a rock. Rocks generally don't go through membranes very well. In fact, they're bigger than the cell, right? So large molecules tend to have a tough time going through this membrane versus a smaller molecule. Let's say they have the almost identical structure, right? But one is much bigger than the other. Then the bigger molecule will have a much more difficult time getting through the membrane than a smaller, similar molecule. Okay, so size here is important, right? Or mass of the molecule is important. The next thing is the molecule polar or not? Right? That makes sense. If it's more polar, it's less likely to go through, right? So a non-polar molecule would have an easier time making its way through the membrane, but a polar molecule would have more difficulty. And then lastly, if it's charged or not. If a molecule has a full charge, it's many orders of magnitude less likely to go through the membrane than if it doesn't have a charge. And so if you have a neutral molecule, right, and I don't mean overall net charge of 0, I mean there are no charges. That's a difference. So not just any zwitterion will do. It has to be a no charge molecule. There are no charges, and they all add up to 0, of course. So that will go through rather easily compared to a similar molecule with a charge. And an example of that is shown at the top right. We have a tryptophan amino acid, and next to it is just the ring structure of tryptophan, which is called an indole ring. Okay, it's basically the R group of tryptophan minus the beta carbon. So would tryptophan or indole go through a membrane more easily? Well, let's apply what we just said. Tryptophan is clearly bigger than indole, so I would think indole would be easier to go through. Tryptophan has polar groups on it, many of them. It's got a carboxyl group, it's got an amino group, it's got another nitrogen, where indole only has the one nitrogen. So again, tryptophan would have a tougher time based on that principle. And tryptophan has 2 full charges, right? And indole has none. So in all 3 cases, it seems like indole would be an easier pass across the membrane than tryptophan is. And it's absolutely right. If you look at our chart, tryptophan falls much farther back on the permeability scale than does an indole molecule. Right? Indole is much more permeable. Right? So those things matter for that one, but what if they're not all easily compared like that? Let's say we compare an indole molecule, which we know goes through fairly well, to something really small, like a sodium atom. I don't mean sodium ion, I mean sodium atom. Right? So sodium with all 11 of its protons and all 11 of its electrons. So totally uncharged. That would go through rather easily. But sodium, as you know, doesn't like to exist as a neutral atom. It would much rather lose its valence electron and become a full shell underneath and have an overall positive charge. So rarely are you going to find sodium in that state. And if you do, it's going to react with something immediately. So sodium will most likely have a positive charge. So it's way down here on the scale. It is the least likely thing to go through this membrane. Glucose would have an easier time going through, and glucose is much larger. Right? So very small charged ions have a very tough time getting through a membrane. In fact, charge is the thing that prevents the most of going through. If I compare a couple pairs or a few pairs of things here, which I'm doing at the practice question at the bottom, and it'll illustrate these 3 principles for you. So in the first one, let's compare an acetic acid and an acetate molecule. Right? So acetic acid and acetate are conjugates of each other, right? The acetate is the conjugate base of acetic acid. It's essentially acetic acid that's lost a proton, right? So which of those two would have a easier time going across the membrane? Acetic acid or acetate?
Acetic acid.
And why is that?
Because it's in its non-conjugate base form, so it doesn't have that negative O minus or that O minus.
Right, it doesn't have the charge, exactly. So they're virtually the same mass, right? Acetic acid has one more proton, literally, than acetate. So it has an atomic mass difference of 1. But that doesn't really make that much of a difference. However, acetic acid, like you said, doesn't have that charge, where acetate will have a full charge. So it's much more difficult to get through the membrane. Right, in the next example, we have acetone or ethanol. Now they're almost the same mass, right? Acetone is slightly more massive, but which one of those do you think would go through more easily, acetone or ethanol? And if you're remembering the structure of acetone, it's just 3 carbons with a carbonyl in the middle. It's the simplest ketone there is. And ethanol is just 2 carbons with an OH group on it.
Acetone?
Acetone would go through much easier because it's less polar than ethanol is. Right? You don't have that OH bond at the end of it that can hydrogen bond and interact with lots of things. Whereas acetone can sort of dissolve a membrane if you add enough acetone to a membrane. Right? It's an organic solvent. But it has a slight polar characteristic on its carbonyl, but not so much so as the alcohol's OH group does. Okay? And then lastly, glucose versus maltose. Maltose, we learned last time, was the disaccharide made from 2 glucose molecules. Which one would go across more easily?
Glucose?
Glucose. Not that glucose is going to go across anytime soon, but compared to maltose, it would have a much easier attempt. And so these 3 things describe the 3 features we were talking about. So the first one describes the effect of charge. Right? A charged molecule has a much more difficult time getting across. The next one describes the polarity difference. The more polar it is, the more difficult it is to get across. And lastly, its mass or its volume or size. And the more massive it is, or the more space it takes up, the more difficult getting across. There's one anomaly in this chart, and that's the water molecule. Right? Water is very polar. It's essentially the smallest possible alcohol. Right? And alcohols don't go across very well. But water is nothing but a polar group, right? It's 2 OH, or it's 1 O with 2 H's on it, so it's like 2 OH bonds, and that's all it is. But it goes through rather well, easier than many small organic molecules like indole, right? And no one can explain why water has such a large permeability in these membranes. Okay? Now there are pores and channels that let water through a membrane, yes, and that's not what I mean. In the absence of any of those, the permeability of water is still very high compared to these other molecules, which doesn't make sense. It's very polar. It should not go across. Okay. And then a quick review of some of these proteins that we talked about before. And I have my cast of characters at the bottom right there. The yellow ones we're going to call the integral membrane proteins. That's the ones that are embedded in the membrane. They have a part of themselves in the membrane. They don't have to go all the way across. Those are called transmembrane proteins. But if a protein has any part of itself, any of its amino acids in the membrane, meaning down into the hydrophobic tail area, then we're going to call it an integral membrane protein. If it doesn't have any of those things in there, like E and F, we're going to call them peripheral membrane proteins. Now D is somewhere in between, right? It's not truly a peripheral protein, because there is a covalent bond to something that is in the membrane, but it's not part of the protein itself. It's that GPI anchor we were talking about, where a protein is attached to a sugar, which is attached to another molecule which has a tail stuck in the membrane, one of these fatty acid tails attached to it. If you look back at the GPI, you'll see what I'm talking about. So is D an integral membrane protein, or is D a peripheral protein? Well, yes and no to both answers. It kind of fits all categories. So we're going to put it in a new category. We're going to call it a anchored protein, or a lipid anchored or attached protein. And so you need some kind of covalent attachment to an anchor, which is usually some fatty acid, and that's attached to, that's not really attached, that's embedded in the membrane. Okay, so if you think of A, B, and C as, again, the members of the band, they're in the band, right? They're in the membrane. And E and F would be the groupies that hang out with the band. They don't play any instruments. They're not in the band, but they're always associated with the band members. And I guess D would be like their manager, right? He's always around, loosely associated with them, but has some kind of contractual obligation to stay in the membrane. Okay. So let's look at these three classes one more time. The integral membrane proteins, that's the band members, the ones that are part of themselves or part of their amino acid chain is definitely in the membrane, right? And they tend to either put just some of themselves or go all the way across the membrane. And even if they go all the way across, sometimes they have bulky regions on each flank of the membrane, right, on each surface. And one of the two ways to get across that membrane is called an alpha helix. The other would be a beta barrel, right, which is a beta sheet twisted on itself. I think we saw a picture of that when we talked about our proteins. But these are the alpha helices that are very long, 20 amino acids long. Some, if you're crossing a bigger membrane, it could be longer, right? But these are long alpha helices, and there's several of them together. In fact, this type has 7, so it crosses the membrane 7 times back and forth, right? So one end amino side is on one side of the membrane, and since it crossed an odd number of times, the carboxy terminus is on the other side of the membrane. But it does go back and forth 7 times. And those 7 alpha helices all bundle together, so it's a 7 helix bundle, right, that holds it in the membrane. And if you remember from the structure of an alpha helix, that all the amino acids, R groups, point radially outward as it turns. So all those radially pointed amino acids, if they're going to interact with these fatty acid tails of the membrane, are going to need to be hydrophobic R groups, right? And if they're interacting with each other, where the helices come together, they could have charge-charge interactions. They could also have hydrophobic interactions. They could have hydrophilics. They could have polars, whatever it might be. It may even have a disulfide bond in it. But they have to face each other and keep their hydrophobic faces outward, and it stabilizes it in the membrane. As far as the peripheral membrane proteins, these are essentially like regular old water-soluble proteins. They may have one face that interacts with another protein or a sugar tree on the surface, or even the phosphates themselves, right, of the membrane. But they don't go in the membrane. They're just loosely associated with the surface of the membrane. Some may have a hydrogen bonding with the phosphates, some may do a charge-charge interaction with the phosphate, and some may interact with the integral proteins themselves. Okay? And the last one at the bottom, the lipid anchored, we said there was a covalent bond to some tether, right? So we have the protein itself attached to some other molecule, like a GPI anchor, which is the sugar, phosphate, and an acetol. And at the end of it is a fatty acid, and that fatty acid tail is buried in the membrane. Now, why does this thing stay there? Well, we call it anchored, because that fatty acid doesn't want to leave the membrane. Then it would have to interact with water. And the tail of that fatty acid was much happier embedded in the membrane. So it's a very ingenious way of keeping a protein next to a membrane, but it's not covalently bound to the membrane. Remember, the fatty acid tails aren't covalently bound to each other, right? But this fatty acid tail is covalently bound to the protein. Okay? All right, so let's look at an example of one of those. Here we have a membrane-spanning protein. It's got 7 of those transmembrane helices. This one's called bacteria rhodopsin. And you have a similar one in your eye. It's just called rhodopsin. And it's the protein responsible for allowing you to see, right? It detects light, right? There's a molecule in the middle of it, which is made from vitamin A, and you can detect photons of light using this protein. Well, bacteria also have this bacteria rhodopsin, and it doesn't really allow them to see as we think of it, but it allows them to detect or use light, right? They can detect light, or they can use the light energy with this molecule. So we have this molecule, this protein, and it moves across the membrane 7 times, and you notice there's some loops on each side, and they make up the interactions with the surface of the membrane. If I were to take that three-dimensional protein and look at its primary structure, there it is on the bottom of the screen, and I just label some amino acids as yellow for hydrophobic, and then the pink or red for hydrophilic or charged amino acids. Okay, so I start looking through it, and I see there's a pattern that emerges. There are long stretches of hydrophobic amino acids. Right now, everyone in yellow is not necessarily completely a hydrophobic amino acid. I agree. We see lots of threonines in there, but those are interacting with the other tails, the other alpha helices. So if you find a stretch of non-charged amino acids is really what we're looking for. No lysines, no arginines, no aspartic and glutamic acids, things like that. And then I say those must be the stretch of the helix going across the membrane. And if I count their length, they're all about 20 amino acids long. So you see 7 of those yellow stretches. There's rarely a aspartate, as this case, in the middle of it, right? And this one had an arginine stuck in the middle of it. This one has an aspartate and a lysine in the middle of it. But overall, those are 7 roughly 20 amino acid in length stretches that form these 7 helices. So could we detect a bacterial rhodopsin-like molecule just from looking at the DNA? Yes. Because if I look at the DNA of an organism, assuming I know it's not going to be spliced in this region, it's easier to do with bacteria because they don't do splicing. And then I say these are translated into this sequence of protein, and I look at the stretch and go, that looks like it could make an alpha helix. It's the right length. It doesn't have any major problems with hydrophilic stuff in it. There's an occasional one, but it could interact with another. And if you look at this, you'll see there's 7 alpha helices in this thing. At the ends of each of these stretches, you notice there's lots of charged amino acids. So at the end of the first one, we have an arginine and a glutamate. And at the other end of it, we have a lysine. Well, all these charged amino acids at the beginnings and near the ends of each of these stretches are interacting with the polar head groups, the phosphates and the cholines and acetols and everything else in the membrane. Right? So it anchors this alpha helix to each side of the membrane as well, so it doesn't move. So all 7 of these put together, this protein stays put where it is in the membrane. It doesn't bob up and down like it's floating. Right? It's fixed in the membrane. Okay? So let's answer our question at the top about integral and peripheral proteins. So which of the two would you think is easier to remove from a membrane? An integral membrane protein or a peripheral membrane protein? And why?
Peripheral.
Peripheral is which one?
Easier to remove.
Why?
Because there is no with the peripheral protein you won't see the hydrophobic interactions occurring in the integral proteins from the hydrophobic amino acids being comfortable with the hydrophobic tails of your phospholipids.
Right. So you can remove
the peripheral is comfortable already.
You can remove the peripheral ones rather easily. It might just be an ionic interaction or hydrogen bonding or maybe a few hydrophobic ones in there. But in general, you can simply add some interfering agent like a salt will generally remove it from a membrane. Right? But the integral one is much more difficult to remove from the membrane, and you need to use something like a detergent to remove it. You really don't disrupt the protein structure. I'd like to keep it intact. And I could remove both easily by just heating it to 200 degrees, and yeah, they're both going to denature. But that's not the point. I'd like to remove it intact. So to remove an integral protein, I need to give the protein a surrogate for the membrane, and we use detergents for that. So we use detergent molecules that could interact with the protein, the integral protein, much like the membrane tails would, but on the other side, I don't have many more copies of those. I have water molecules that interact with the detergent. So in the right concentrations, you can remove an integral membrane protein with a detergent. If you add too much, you may denature the protein. If you add too little, you don't get it out of the membrane. Right? So you got to work on your concentrations to get those removed. And then you maybe could do some experiments with that protein, such as crystallography or some chemistry, or even enzyme kinetics if it's an enzyme. But the integral ones are much more difficult to get out of the membrane. Okay, some other ways across the membrane would be a beta strands wrapped around themselves to make this sheet that forms a tunnel, right? And it's called a beta barrel. Now the ends may look like they're open, but they could or could not be open depending on all those loops you see. All those loops could close the ends of this molecular tin can, right? So it has a pore in the center, but it also has lots of amino acids on these loops that could seal off the tops and bottoms of this can. And even in the interior, you may have some loops that move all the way across and seal the interior. So it may not be a perfect cylinder that goes through, but it has a cylindrical shape. Okay? And if I look at its structure in the tertiary there, I see that cylinder shape. Translate that to the primary structure, just give me the list of amino acids again. I won't find long stretches of hydrophobics like I did with the alpha helix. Because here, the way a beta sheet works is, remember, they're lining up side by side, and all the amino acids are pointing towards one face of the sheet and then towards the other face of the sheet. Back to the first and then to the second. And it alternates faces of the sheet. So again, you can take your fingers and line them up as if they were the strands of a beta sheet. And then if you look as you're going down any one finger, the subsequent amino acids are facing above and below your hands, above and then below, above and then below. Even if we turn it into this twist, we say there's one amino acid will be pointing inward and then outward, inward and then outward. So what we'd like to see in this type of pore is every other amino acid roughly, it's not exactly every other one because we have some turns and some twists in the sheet, but roughly every other amino acid is a hydrophobic one to interact with the lipids. And every other one is pointing to the interior, and it's going to be hydrophilic to interact with the channel that's going through. And if you look at the primary structure there, it's not exact, but if you look overall, virtually every other spot is a hydrophobic spot. Sometimes there's two in a row, sometimes there's not, but it depends on where the turn is in the structure, if it's near the surface of the interior. But all of these have little loops in between, and the horizontal slanted lines are the ones that are interacting with each other with hydrogen bonds. Right? So this is how you detect a beta barrel type protein from the primary structure. Look for roughly alternating hydrophobic, hydrophilic amino acids. Where if you see a long stretch of hydrophobics flanked by polar ones or flanked by charged ones, it's more likely to have an alpha helical structure. Okay. So let's see how one of these works. So we're going to do an example. This enzyme is called prostaglandin H2 synthase 1. Right? So it's a really long name. But all you need to remember about it is it's one of your enzymes that's involved in the inflammation response. Right? So inflammation does not mean infection. Those are not the same thing. Inflammation means the influx of inflammatory cells, like white blood cells. Okay? And so this molecule is responsible for ringing the bell and telling them, come here, right? Come to this location. So it makes a very small signaling molecule called prostaglandin H2, which we're not going to describe here. But that molecule attracts all those white blood cells. It's a very short-lived molecule. But the prostaglandin H2 molecule, which is the signal, is made from the lipids of the membrane, right? This is one of those molecules with multiple carbon-carbon double bonds in it, like our arachidonic acid you saw last time, right? With all four of its carbon-carbon double bonds. And they can fold up and make very short-lived, unstable molecules that act as signals for white blood cells. But the raw material for making it is one of our lipids. So the enzyme, prostaglandin H2 synthase 1, shown on the left there, it has to be situated next to its raw materials, next to the membrane. That's where from it gets its raw materials. So is prostaglandin H2 synthase 1, that's the protein shown on the left, a peripheral membrane protein, integral, or lipid anchored, based on the picture?
Lipid anchored?
Everyone always says that. It's actually integral. Does it have part of itself, meaning part of its chain, its amino acids, in the membrane?
Yes, it does.
Yes, so that's the part shown in orange here, right? So the blue part is outside the membrane, but that's not the entire protein. This is also a finger of the protein, right? So it's like it's got its hooked finger in the membrane, right? This is not a lipid anchor. This is not an attachment to some other part that's embedded in the membrane. It's part of the protein chain itself, right? And you notice it's full of hydrophobic side chains. And once it does fold up and puts this finger of helices in the membrane, it's stuck there. I'd hesitate to say anchor there because it's not a lipid anchor, but let's just say it's glued there, right? This link, this loop of hydrophobic chains of amino acids here tend to keep this protein associated with the membrane. Okay, so what it does is grabs one of these lipids from the leaflet here, right? Cuts it off of the phospholipid, and it channels it through the protein itself. And as it's coming through, it folds it up into the prostaglandin that we're going to release as a signaling molecule. On its way through, there's a hydrophobic channel, which makes sense because we're making a hydrophobic thing into something more water soluble. So there's a hydrophobic channel, and on the way out that channel, there's kind of like a gatekeeper or a bridge attendant or something like that, where it's a certain amino acid here that's normally not in the way. It's serine 530 over here, right? It's just this OH sticking out there. But if I were to attach something to the serine, then it would block this channel, right? And this is what aspirin does, right? So when you take aspirin to reduce inflammation, it's one of your non-steroidal anti-inflammatory drugs. So take aspirin, and aspirin is an acetate group stuck on the side of a salicylic acid. So it's called acetyl salicylic acid. And what it does, if it reaches this area, is this molecule's ester bond is broken by the serine. So the serine sitting into position in the beginning of this channel, or technically the end of the channel, but the channel at the surface, this serine's OH attacks this ester bond, right? So the O attacks the ester bond, acetylating the serine and releasing the salicylic acid, right? And it's just degraded as a byproduct. But now I have an acetate group stuck on this serine. Is it on there permanently? No. It's again an ester bond, but it's in a hydrophobic area. So it's very unlikely that a water molecule can get in and break it. It takes time, right? It will do it eventually, but it takes time. So this will slow down the production of my prostaglandin H2 that this enzyme is making. And if you take a lot of aspirin, it's going to slow down almost all of these enzymes, right? So the dosage of aspirin will change with the amount of pain that you're having. And if there's more prostaglandins being made because inflammatory cells need to go here for some reason, this will decrease that inflammatory response. So aspirin also has the side effect of, since it's decreasing the inflammatory response, it makes you more susceptible to infections because you're lowering your immune response. Okay, so you got to be careful how much you take. There's other side effects such as the excess aspirin. Most of it doesn't make it past the stomach because this ester bond gets cleaved rather easily, right? But this is how aspirin reduces fevers or inflammation response. There's 4 hallmarks of inflammation, if you guys remember those, right? There's the fever or the heat, right? There's the redness, there's the swelling, and then there's the pain, right? It's easier to remember them in Greek, and if you want to look up the Greek names, you can, but I won't talk about them here. Okay, so this is an example of an integral membrane protein and how it does its chemistry, but it can be blocked by a molecule such as aspirin. So let's talk about the membrane as a whole and what components make up the membrane, right? So there's 3 main components. There's the lipids themselves, there's the proteins that are embedded in the lipids, and there may be some sugars around, the carbohydrates. Okay? There aren't going to be any nucleic acids in the membrane, right? Those are going to be inside the cell or in the nucleus. So let's look at these 3 components and think about the simplest membrane we can think of in a biological setting. Let's go to a simple bacterium. So at the bottom of the chart here, very bottom of the list, is our halobacterium purple membrane. So a very simple bacterium that lives in a saltwater solution. So if you look at its membrane composition, it's about 3/4 protein and 1/4 lipid. An easier way to think about that is the lipid bilayer is just chocked full of protein, right? It's as much as you can stuff into the membrane. Now, why is that? This little bacterium, its outer membrane is its only membrane. So it must perform all the functions that membranes need to perform. Moving things in and out, doing respiration, doing the electron transport chain, doing ATP synthesis. Everything this bacteria needs to do occurs at this one membrane. Of course it's full of protein. It's got all these components in it, right? It also has no carbohydrates in its membrane. I wonder why. There's no carbohydrates. So what must be the point of the carbohydrates on the membrane? Well, we'll see when we get to some with carbohydrates. Let's look at the next one up. How about a spinach chloroplast membrane? So this is a plant, but inside of the plant there's a chloroplast, so it's an intracellular organelle, right? And it's roughly the same. It's mostly protein, again around 70%, lipid about 30%, and a few carbohydrates on there. So it looks a lot like a bacterium, which you'd expect. If you look at the mitochondria again, we have roughly 75% protein again, a quarter lipid, and just a few carbohydrates. So again, mitochondria look a lot like bacterial membranes. If we move up to the top of the list, where we have human and mouse, and we're down to roughly 50% protein and 40 to 50% lipid. So far fewer proteins there. But the number of carbohydrates has gone up greatly, right? We're up to 8% carbohydrate now. So what must be the point here? Human and mouse plasma membrane cells, these are not their internal cells. This is the external cell membrane. Doesn't have to do all the things that the bacterial cell membrane had to do, right? It doesn't have to do the ATP synthesis. It doesn't have to do the electron transport chain. It doesn't do photosynthesis in the case of plants. So it does all these other functions like just moving things in and out, maintaining a gradient of sodium and potassium and so forth, but it doesn't need all the other functional parts because they have organelles that do that for them, thus the drop in protein content. But it does have an increase in carbohydrate content. The reason being, if you're a halobacterium, if I run into another cell, it's competition. It's the enemy, right? It's competing for food resources. I don't need to be told what's on its cell surface. It's not me. However, if you're a human cell or a multicellular organism, I don't want to assume every other cell I run into is competition because then you won't be a functional multicellular organism. I need to identify my comrades. So you have 8% carbohydrate there. Those are your sugar trees on the surface, so that one cell running into another knows, oh, you're part of our organism, right? You're part of us. Let's not destroy you. If you don't have the right carbohydrate tree, then we're in trouble, right? And inside the cell, the mitochondria and the chloroplasts don't need a lot of carbohydrates in animals because they never encounter other membranes. They never encounter another cell. They're intracellular. In plants, they do. They encounter other organisms inside the plant cells. Those are the little rhizobiums in the plant roots that actually are intracellular, not so much parasites, but it's a happy relationship between the plant and these organisms. And eventually they will become another organelle, give it another few million years. But what I want you to get out of this chart is that the protein content tells you a lot about the enzymes, about the membrane's function. Right? If it has a lot of protein, it must do a lot of things. If it doesn't have so much protein, like in the human plasma membrane, those functions must have been put off on other areas or other membranes. And the carbohydrate content will tell you, does it need to recognize or identify other cells as friend or foe? So some other characteristics of the membrane are these balls and sticks you see here with the tails and the phosphate head groups. They're not stationary, they move, right? And when I say they move, I mean they slide past each other, right? They're not bound to one another, no covalent bonds here. It's just that hydrophobic association. So they do slide past each other, right? And so we call that lateral diffusion, right? So one leaflet has a phospholipid there sliding past all the others, and they move rather rapidly. It's a relative scale here, so 2 microns per second for us seems exceedingly slow. But from this molecule's point of view, that's pretty fast, because 2 microns is much larger than its own diameter, or own distance, or own length. So it's moving rather rapidly from its point of view. However, the transverse diffusion, so one phospholipid flipping from one side of the membrane to the other, is very rare, right? It only occurs once every several hours or so. So it's a very slow occurrence, right? That's what they mean by slow here. Not so much speed, but it doesn't occur frequently. This harkens back to our enzyme definitions for slow, right? And then transverse diffusion from one side to the other doesn't occur very often, but it has to occur, right? Because these phospholipids are manufactured by the cell in the or in the cytoplasm, not the nucleus, but in the cytoplasm. And when the cell grows and divides into 2 other cells, they must be put on the outer leaflet somehow, right? They can't find it outside. You made it in the cytoplasm. So there must be a way of putting the phospholipid from one side to the other. And there's an enzyme aptly named flipase that does this. But moving across on its own by diffusion is very, very rare. Right? Whereas moving laterally happens all the time. They're always sliding past each other. This is what we mean by a fluid membrane. Okay? Proteins, however, the ones that are embedded in the membrane, never flip over. Right? They don't have transverse diffusion. Now, proteins could be purposely turned over by other enzymes, but proteins don't spontaneously flip over in the membrane, have one face flip to the other side. It doesn't happen. Okay, so here's an experiment showing that. If I take a leaf cell, right, this isn't the whole leaf, it's just one cell, and I stay away from the nucleus, and I photo bleach it, as shown from panels A to B. So I use a laser to photo bleach the area so it loses all of its fluorescence, right? So all the molecules that are there no longer react to the light being exposed to them. And then I let it recover. I give it time, right? So and over time, I notice that area regains its fluorescence, right? Or in this case, in the diagram here, it shows it by a green color, right? They don't really lose their green color. It's just a diagram, but they lose their fluorescent ability. So if I measure this on a graph, and that's the graph D at the bottom, if I take the initial intensity of the fluorescence of the static leaf cell, you notice it was constant. So over time, it's showing a constant measurement. Then I use a laser to photo bleach it. It instantly drops because I've bleached all the fluorescence there. It's not chemical bleach, it's a photo bleaching effect with a laser. Then if I wait a while, over time, that fluorescence, shown as the green color in the picture, recovers. Eventually we'll reach the normal levels again. But this curve should have a meaning to you. This shape means something to you. What does this mean? What shape does this curve have? Think back to when we did enzymes.
Hyperbola?
Right. It's a rectangular hyperbola, exactly. And if it's a hyperbolic function, eventually it reaches what? What do you call this line that it reaches?
Asymptote.
It's an asymptote, exactly. Now it could mean Vmax in the case of one of your graphs, and it could mean equilibrium in the case of the other. In this case, we're looking at basically product over time, right? Because I'm measuring fluorescence as my output. So when it finally recovers and reaches that asymptote again, we're back to equilibrium, right? So this is a recovery of the color. How is it recovering? Well, these molecules are moving side to side, right? Along one leaflet and side to side along the other leaflet, this lateral diffusion. So some at the edge are moving into the area we bleached and some in the area are moving away. And since there's a large volume of them, a large number of them, not just the area we bleached, this is grossly exaggerated, the area that we get bleached, eventually they'll move through here and diffuse enough that it regains the color. Okay, so what I can do is, I know if I measure the fluorescence over time, this will slowly approach this asymptote, but to measure how fast these things move, I want to catch it when it's first moving. So I measure the initial velocity again, just like we did with enzymes. Right? So I'll measure the speed, which is the slope of this line, as how fast these things are moving. Right? So my change in intensity over time gives me an idea of how fast these molecules are moving sliding past each other in lateral diffusion. And that's where we get this measurement of about 2 microns per second. Okay. Right, so the other thing we can say is, what about the lipid tails themselves? So someone was asking this question earlier. If you have all saturated tails versus some with some cis double bonds in them, right, they can't pack together as well in the latter case. Whereas in the former case, they pack together very well. Their melting temperature will be much higher, right, than it is in panel B. It'd be much lower, easier to melt these. So if I measure this membrane, right, it's many, many of these put together over time, and I measure, is it more of a solid membrane or is it a more fluid membrane? Are things moving? And I can use the previous experiment to test that. Because if I photo bleach it and I wait a really long time and nothing recovers, it's solid-like, right? It's not moving. And if I do photo bleach it and things do recover, it's more fluid-like. And the faster it moves, the more fluid-like it is. I do those measurements at different temperatures, and I'll get a graph that looks like this. And it has this sigmoidal shape, right? It doesn't suddenly become solid to liquid state, right? It's not a single molecule melting. It's not a melting point in that sense. We call this transition temperature or melting temperature, at which the membrane as a whole becomes more fluid-like. This is much like when we talk about melting DNA as well, right? When the DNA strands come apart, we call that the denaturing or melting temperature. It's not a single point where all the molecules suddenly come apart. It's where half the molecules have come apart. Okay? So what this means is if I have a membrane made of these materials, this either all saturated or maybe it has some unsaturation in it, and I measure its solid versus fluid-like recoveries, and over time I get this plot. The point at which this 50% fluid-like, 50% solid-like occurs is called my melting temperature. That's our definition. Okay? So what do you think the melting temperature is in the membrane made of molecules like in A, where they're saturated, versus membrane made of some like B?
I think the melting temperature for A is going to be a lot higher than the melting temperature for B.
Right. Do you think the shape of the curve will be the same?
Yes.
I think the shape, yeah.
Yes.
But where that TM value lands.
Right. So instead of moving the TM, let's leave it like it is and grab this. If you could imagine grabbing the whole picture and just sliding it left or right on the temperature scale, that's what it does with these measurements. Or with these molecules. With the all saturated one, it could look like this, we can imagine. And with the sites of unsaturation or cis double bonds introduced, we can slide the whole scale down this way, right? This whole curve would be sliding to the left. At a lower temperature, it would melt. Okay? So it's just a graphical way of explaining what you saw last time when we talked about lipids and their melting temperatures. We're just doing it on a scale of many of them now, not just one, but many of them in a membrane setting. Okay? So we have a practice question that I used to put on exams, and I just use it as practice in class now. But there are fish that live in desert streams, because you remember in the desert, it's very hot during the day, and it's very cold at night, because you don't have the atmospheric above it to moderate the temperature, and there's no water other than these small streams to modulate the temperature near the surface as well. Right? So no humidity in the atmosphere. So certain fish in these streams alter their metabolic composition when they transition from day to night. This is very unusual. Most organisms don't do this. So we go from day to night. What do they do to their membranes?
You switch from saturated membranes to unsaturated membranes.
So they put in some extra saturated tails and remove some of the unsaturated tails. Or let's just say they can reduce some of the double bonds to make them saturated. As opposed to going from day to night or night to day, we have the opposite changes, right? When we get to the heat of the day, we want more saturated ones because the day is going to heat up and membrane needs to stay fluid. Whereas at night, we can't have all those saturated ones because it'll turn into a solid. So we need to introduce, as the sun is setting, introduce some double bonds in there or replace the fatty acids with double bonds. Fatty acids with double bonds in them. So it's a very unusual way of maintaining the fluidity of your membranes, but they've evolved to do this in this desert environment, right? It's very unlikely they have conscious control over it. That would be amazing if you could, but it does it in temperature drops or temperature rises. It triggers this change of certain enzymes. Okay. One last thing in the membrane would be our cholesterol molecule, right? You remember last time you were learning how to draw it with its 17 atom and 4 fused rings of its sterile core, but there's one OH group on carbon number 3, and I said it sits in the membrane like a bobbing cork. So that's the cholesterol molecule here. This is exaggerated, the size of the OH, but the OH has the only polar part of this molecule. It's going to interact with the polar head groups, the phosphates and the alcohols. And then the bottom part of the cholesterol molecule, including its tail out here on number 17, and the core structure are all hydrocarbons, right? They're not going to interact well with water, so they interact with the long tails. What effect does this have on the membrane? Well, it has 2 main effects. And I think I mentioned these last time as well, but I'm going to make sure I state them again. So if the Membrane is fluid, and we raise the temperature, at some point the membrane will fall apart, right? Having cholesterol present will raise the temperature at which that occurs. So it keeps the membrane intact at a higher temperature. It does this because the cholesterol molecule with its fused rings is very rigid. Things can bind to it and hold on for stability, right? The tails aren't flying everywhere. Something solid, something rigid that won't move and won't bend because of all these fused rings to hold on to. On contrary, at the low temperatures, all these tails are trying to compact into a solid, right? And if they have double bonds in them, it's a more difficult task. But if they're all saturated, it's rather easy to do, unless there's cholesterol around. So this bulky cholesterol molecule gets in the way of these tails packing tightly. They have to go around it. It does not bend, right? So it's very bulky. The tails have to make their way around it, and thus the point at which the membrane actually freezes or becomes a solid has been lowered. So in both cases, cholesterol has widened the range above and below the temperatures at which it would become unstable or become a fluid falling apart or fluid freezing on the two extremes. So cholesterol widens the fluidity range of the membrane based on temperature. Okay. Also, you'll find cholesterol is associated very readily with those sphingolipids we talked about. So replace the glycerol with our sphingosine, and they tend to occur in clusters. They don't just happen diffusely through the membrane. The sphingosines tend to occur in groups and stay together. And those groupings of sphingosine lipids, or sphingosine-based lipids, as opposed to the glycerol-based ones, have cholesterol at a higher concentration near them. Right? They tend to stick to them. And we call these lipid rafts because the lipids that are higher in concentration of sphingosine in that region tend to have more cholesterol associated with them. And this is a player for certain proteins might bind to that, certain cofactors like to bind to that, and it has certain properties that the membranes without the sphingolipid and cholesterol groups don't have. The last thing I want to talk about for this protein part is the channels that can be made from these transmembrane proteins. So if you have a transmembrane protein, it goes all the way across. Perhaps there's a channel that goes through the center, and we can let certain size molecules go through this channel. It can depend on their charge. It can depend on their shape, geometry. It can also depend on their mass, right? So, and shape. So if it's a really long protein, it could slither through this channel. That's how we export some proteins. Or it could be only letting single atoms through, like an ion channel, like only letting sodium through, or only letting potassium through, or only letting chlorides through, right? So this channel may be open all the time. It may be closed all the time until something comes along and opens it, such as a ligand binding and opening a flap on the surface. It's called a gated channel. The flap may open when a molecule binds on the outside, like a hormone from another cell. It could be when a molecule binds from the inside, so the cell sending its own signal to the cell surface. It could be an electrical potential, right, where the voltage changes and the channel opens. So there's lots of options for opening these channels. In fact, you could have one open with light shining on it. Okay? So there's lots of ways to gate these channels, and there's lots of things they could or could not pass through the channel. Right? And we'll look at an example of that. Okay? Before we look at our example, I want to briefly remind you of something that you probably had in high school or freshman biology class, or even maybe in a chemistry class. And it's these simple terms called passive transport and active transport. And what I mean by that is passive transport does not require any input of external energy. Okay? It happens on its own, doesn't need any input. Active transport needs an input of energy, and the reason being is generally moving a molecule or a thing up its concentration gradient, right? Whereas passive transport always goes down its concentration gradient. They're very simple definitions. So passive transport means a molecule is moving from an area of high concentration to lower concentration across some barrier, like a membrane. And in active transport, it's going from an area of some concentration to an area of higher concentration relative to the first place, again across some barrier. That's the difference between passive and active. We can also have what's called simple and facilitated. Simple means it goes across on its own, right? So like an indole molecule can just go right through the membrane, no problem. Sodium atom, not so lucky. It can't go across the membrane. So a sodium atom going across would have to be facilitated. It needs something to carry it across, like a pore or a taxi or a channel or something to get it across the membrane. However, if it's a water molecule or an indole molecule or something like that, it's really small, can go right across on its own. That's called simple. Right? It's often termed diffusion. Okay. The last thing on the page here is in the active transport category, which by definition cannot be simple. It must be facilitated. Nothing goes up its gradient without help. Right? I know it requires energy, but something has to use that energy, some pump. Right? So you can't have a molecule going against its concentration gradient without some input of energy and without some way of facilitating use of that energy. So you can't have simple active transport. It doesn't make sense. So active transport is by definition facilitated, and it can occur in two varieties. We have primary active transport and secondary active transport. Right? There is no tertiary and quaternary or anything above that. What we mean by primary active transport is it uses the power source directly, such as the hydrolysis of ATP, or the hydrolysis of UTP, or a photon of light, or some direct power source is primary active transport. What secondary active transport is it takes the results of a primary active transport's work. Say our primary active transport pumps protons across the membrane. Our secondary active transport then allows those protons to come back across and at the same time transport something else across the membrane against its gradient. That may be in the same direction, it may be in the opposite direction. But what secondary active transport means is it's secondary to some other input of energy at a primary site. So if I, for example, use light energy to pump protons across a membrane, I'm just making up an example. That makes a gradient of protons. Those protons want to come back across the membrane. So at another site, we let them come across. But as they come across, they must bring a chloride with them, let's say. Just making up an example. That pump of chlorides across the membrane against their gradient by use of the proton gradient is a secondary active transport. Now if I use that chloride gradient that I just made to pump, say, something else across the membrane, maybe magnesium or something, it's still called secondary active transport because it's still dependent on some primary source. Everything after the primary source is called a secondary active transport. So let's look at an example here of active transport. So on the right is a diagram of a membrane again, and we have some transmembrane protein sitting there. It happens to be an enzyme. It's a pump, and it's called the sodium-potassium ATPase pump. Okay? What it does is it pumps sodium ions out of the cell and potassium ions into the cell. And in both of those cases, those are against their concentration gradients. Okay, so it pumps 3 sodiums out and 2 potassium ions in at the expense of an ATP hydrolysis. Okay, so it's powered by ATP hydrolysis, and in 3 sodiums, there's more sodium outside the cell than inside normally. So pumping the sodium out is against the gradient. That's going to require some energy input and a facilitated pump like this. And pumping 2 potassiums in is also against its gradient. So there's generally more potassium inside the cell than there is outside. So using ATP, we make this exchange and make the gradients even more great of a gradient than they were before. Right? So the cell maintains this by using this pump, and about 1/3 of ATP in a resting cell is used to maintain these gradients. Because we're going to use the sodium and potassium gradients created by this to power other things in our secondary transports. So the 1/3 of the ATP consumed in a cell is just to maintain this gradient. And if you look at a neuron, it's about 65-66% of it is used, or 2/3 in a neuron is used to maintain this gradient, because neurons are very sensitive to their electrical potentials, and that's how they function. This also controls our cell volume because the water will go with the excess charged ions. So as the sodium goes out, 3 go out, 2 potassiums go in, water is going to want to follow the sodium out. Okay? And also this gradient we're creating and maintaining is going to be used in a secondary active transport to move other molecules across the membrane. So what if I had a molecule that would inhibit this active transport pump, this primary pump? That wouldn't be very good for the cell. Right? And there's a molecule called Digitalis, it's from the foxglove plant, that inhibits this very pump. right? And if you inhibit this pump, the cell will die, right? If you inhibit all of them. So when you take Digitalis as a cardiac medication, it's not in high concentration, right? And what it does is it slows down this pump. It doesn't shut it down completely. It does inhibit the one it binds to, but there are many of these. But it slows it down enough so that the concentration of potassium inside is a little less than we want it to be, and the concentration of sodium pumped out is a little less than we want it to be. So the cell can't put the calcium that's normally in the cytoplasm back into the ER or the SR in the sarcoplasmic reticulum in muscle cell. And since it can't put it back in because we don't have the gradient we need here, the concentration of calcium tends to stay a little higher in the cytoplasm, which makes the muscle contraction a little stronger. But if you do this too much, you'll kill the cell. Okay. Right, so let's look at some of these other transporters that make use of this. So at the top here, we have what's called an ABC transporter, and it stands for ATP binding cassette. It's basically a pump that uses ATP as the power source, and it pumps various things. Right? The one we just saw was sodium and potassium were being pumped. Here, this one pumps a variety of things, and bacteria have these things. They're ABC transporters. What they do is they pump molecules identified by the cell as what shouldn't be in the cell. If it has no use, it pumps it out of the cell, get rid of it. So if you try to kill the cell with certain antibiotics, that the way they work is they inhibit the ribosome, for instance. They have to get inside the cell to work. So when they get in the cell, the bacterial cell, if it has these multi-drug resistant proteins, these ABC transporter pumps, they'll identify the molecule as foreign and pump it out of the cell. But it doesn't go away. You didn't destroy it. And if it gets back in, we'd have to pump it out again. So those cells are spending a lot of time trying to get rid of the drug that could kill them. And that also slows down their growth, giving the immune system a chance to help. A modification of that, because these multi-drug resistant proteins pump out large numbers of things, right? They don't really specify one particular molecule. There's a variety of molecules they can pump out. Whereas at the bottom, you notice we've had the same basic sequence there, if you look at the primary structure sequence, but that one white piece in the center is a little bigger, right? It encodes for a little more feature, right? More amino acids there, represented in the diagram by the sphere in the middle of the connection. But otherwise, all the parts are the same. The ATP utilization part, the part that does the physical pumping. But because of this little addition in the center, it's very selective about what it pumps. And this is a calcium channel pump, right? It won't pump anything else. So by putting the selectivity filter in here, right, it's a modification of this non-selective pump, I can select for only calcium ions being pumped. Right? And problems with the former example here, if a bacterium has a problem with this multi-drug resistant protein, they're going to be very susceptible to many drugs. Right? So if we inhibit this pump with a different kind of drug, it makes our other drugs more effective. Right? That's a synergistic approach to antibiotics. And in the bottom, if we inhibit this calcium channel, perhaps because it has a mutation, then we can't pump chlorides across, and that's going to lead to an inability to move fluids around, and that's one of the leading causes of cystic fibrosis. Right, so secondary active transport. So we've built up a gradient of, say, sodium and potassium at our primary site. Then we're going to utilize that gradient, but to pump something else. Right, so if I'm pumping something else, let's say in this example at the bottom, sodium is being let back in the cell. Right? Remember, we pumped it out at the primary side on the left. We're letting it back in the cell on the right. And we say, if you come in through this little transporter, you must bring a date. You got to bring a glucose with you, because this thing only lets a sodium with a glucose back in the cell. Well, again, glucose would be going against its gradient here. There's clearly more glucose inside the cell than outside, and there's more sodium outside than inside, so there's impetus for the sodium to come through, and it drags the glucose along with it. So this is a secondary active transport. Yes, it's pumping glucose against its gradient, but it's using a gradient created by another primary source, our sodium potassium ATPase pump. Right? So this is secondary to that pump, without which it wouldn't function. Since it's sending glucose and sodium across the membrane in the same direction, we're going to call it a symporter. They're both going in the same direction. Had it done something else where as it lets a sodium in, it lets, say, another molecule out, it would be called an antiporter, since they're going in opposite directions, and those also exist. So our last thing we'll talk about is a very specific one, is a sodium and potassium channels. So this sodium channel that we're talking about, letting sodium in and out, if you block it, that is lethal to the cell, right? If you can't maintain the sodium in the cell at a certain concentration, if you block the channel, then the cell is going to die rapidly. And the most sensitive cells to this are going to be nerve cells. Okay? So this little fish in the center is got a lot of spines on it, and it's a delicacy in Japan to eat the meat of this fish, but it's very dangerous to prepare it. So each of these spines has about a milligram, right, of this the tetrodotoxin you see on the right. You don't have to memorize its structure. But this molecule on the right efficiently blocks sodium channels, and only sodium channels, right? It's very selective. And if you have this molecule, if it gets inside the cell or near the cell membrane and blocks these channels, those cells will die, right? And a lethal dose in humans is about 10 nanograms, right? And so there's roughly 100,000 times that dose on each spine of this fish. So if you get stuck, you'll be dead before you can think about, oh no, I was stuck, right? It kills the nerves that fast. So if you're preparing this as a delicacy and a meal in Japan, and your knife happens to hit one of these spines and you get it into the meat, you're going to kill millions of people with this dish if you serve it to them. So when the Japanese chefs prepare this, before they serve any of their preparations, they must try it themselves, right? So I could imagine training for this is rather tedious before you can actually work with the actual fish. Okay, so here's an easy question for you at the bottom. So if I have a channel that is let's say sodium channel, like the one we just talked about that fish blocks, or the fish's toxin blocks. If I have a potassium channel, and I'd like to let a molecule through it. A sodium will go right through, right? This is the sodium channel. So a sodium will go right through the channel. Why will a potassium ion not pass through that sodium channel? Well, I'll give you some idea of how big the sodium and potassium ions are on the left here. The sodium ion, right, the gray sphere in the center represents the ion after it's lost its electron, right, valence electron, is 102 picometers, which is 0.102 nanometers, or 1.02 angstroms. So effectively a little more than an angstrom, right? So this is the width of a sodium ion, whereas a potassium ion is about 1.38 angstroms. So quite a bit larger, right? 30% bigger than the sodium ion. So why would a potassium ion not go through a sodium channel? That's an easy question. What do you think?
Too big.
Yeah, it's too big. Of course, it's too big. The channel's only so wide, and it will not let it through. All right, here's another question. Why do sodium ions, which are smaller, not pass through potassium channels? And they don't. You can't say they're too small because that doesn't make any sense, right? So what's the reason why a sodium will not pass through a potassium channel? Anybody have a guess?
Would it be because the sodium doesn't cause or doesn't interact with the channel in a way to cause a conformational change for it to allow to pass?
Well, it's not so much a conformational change, but the interaction part is right. So if you look at the diagram of the channel there, this channel, if you think about it, this is a typical potassium channel, doesn't have to be very narrow all the way across. It's much like the Panama Canal, right? Most of the Panama Canal is a wide lake and waterway, right? It's not very narrow at all. But where it has to go through the locks to pass from the canal itself into the Atlantic or Pacific Oceans, it's got to be very narrow to control the passage of the water. So this channel only needs to be as narrow as any one point to select for what goes through it, right? Much like the ships that go through the channel there. If a ship is bigger than the narrowest point of the passage, it can't go through. The same thing here. If an ion is wider than the narrowest point, it can't go through. And that's how potassium can't go through sodium channels. However, we know sodium is smaller than potassium, so how do we prevent it from going through? Well, there's a region right near the entrance there, much like the Panama Canal, where you have to get through and interact favorably with the things lining the channel. Okay, it's 3 angstroms wide, so it's twice as wide as a sodium atom, or 3 times as wide as a sodium atom, twice as wide as a potassium ion. So how can it easily select from one or the other? Well, here's the way it works. If you have a sodium or potassium ion, they're not floating around as just the ion itself, right? It's going to associate with water molecules. So those potassium and sodium ions, as you see in the bottom here, are surrounded by a shell. It's called a hydration shell of water molecules. In the case of the potassium on the left at the bottom, we have 8 water molecules associating with the potassium. Right? So the oxygens are going to orient themselves toward the charged potassium, and the hydrogens are going to orient themselves facing out to other water molecules. And in the case of the sodium, the geometry dictates that it's going to have 6 water molecules facing it. Okay? The number of water molecules is not the important thing here. It's the size of this hydration shell. Right? So can you imagine the sodium was only 1.02 angstroms wide, but with these shell of water molecules around it, it's much bigger than any channel it might go through, sodium or potassium channel. It's way too big. It's several angstroms wide. Same thing with the potassium ion on the left. It's got 8 water molecules around it, but it's much bigger. It's several angstroms wide. It will not fit through the channel. In order for these ions to go through these channels, they must first shed their water molecules. Right? They got to take all the water molecules off, jump in the channel, right? To remove the water molecules from the charged ion is not easy. You notice we look at our scale again here on the left. To remove these 8 water molecules from a potassium ion means we go up in the energy scale or up in the instability scale. But if that potassium ion then jumps in a channel like the one here on the right at the top, right? The potassium ion, instead of associating with all these water molecules, can associate with the oxygens from the amino acids that are lining the channel. Right? These are the backbone carbonyls lining the channel. So they substitute, they're surrogates for the hydration shell. And in fact, if it jumps in the channel, it's actually making connections with these, which is a more stable thing, so we lower the energy of this interaction. And it actually is less energy, it's lower than it was with the water molecules around it. So because I start here, I go up and I end here, this is more favorable than it was in solution. Right? So it's favorable for a potassium ion to shed its water molecules and jump in this channel. Okay, so let's do the same thing for sodium. The sodium, to lose its water molecules, is a huge investment of energy. Right? And if it were to try to jump in this channel, sodium is not wide enough to interact with both sides of the channel. If you imagine potassium is a human jumping in, and we reach out with arms on each side and legs on each side, and we hold on to something on the wall, right? Whereas the sodium jumping in is like T-Rex, right? The arms aren't long enough to reach the wall, right? So it can't hang on with all of its limbs. It can only make 4 connections. It can't make the other 2. So that doesn't give you enough of a resolution here, right, to resolvate it in the channel. This is not favorable. It goes uphill. Whereas the potassium entering a potassium channel is actually downhill. That's why it hops in the channel. Well, that's great if you want to get a potassium in the channel. That's wonderful. How do you get it across now? Because this was very favorable, it doesn't want to leave. It wants to sit there. So what we do is, if we have one potassium shown at the top here, shed its water molecules and hop in the channel. Here it is in the channel, the second pane. Right? Then another potassium molecule, it gets near the entrance. It would also like to enter the channel. And because it's also positively charged, it pushes this potassium down to the next set of carbonyls. Right? It sheds its water molecules, hops in the channel as well. These 2 potassiums next to one another, again, not favorable. One moves down even further. Right? And this process continues where we have more potassiums line the channel or hop in the channel lined by these carbonyls until one is expelled from the other side. Right? And then more can continue to come in the first side. And this will continue as long as it's going down its gradient. Okay? So the reason that sodium cannot enter this is because it doesn't make favorable interactions with the carbonyls lining the channel. Not because it's too small to enter, but it's shedding of its water shell and then reattaching itself to the carbonyls is not a favorable energetically thing to do. Whereas the potassium, it's a slightly favorable thing to do. I don't want it to be very favorable because then I'll never get it to leave the other side. Okay. So to recap, we have channels that are gated, some are not gated. They can be selected by photons to open, or ligands binding to open, or a small molecule inside the cell binding. We can have passive transport, which is downhill, right? And only it could occur with or without the help of something. Could be simple or could be facilitated by a taxi or a channel. We could have active transport, which is generally against the gradient and always requires to be facilitated. And it needs some kind of energy source. If it's a direct energy source, like a ATP or a photon or some other high energy molecule that gets broken in the process, it's primary active transport. If it's taking advantage of that in a secondary interaction later, we call it secondary active transport.